right, welcome back, everybody. I hope you got some good sleep. We are going to start today with an introduction to metagenomics. This is sort of the basis of what we're going to build on in the course here today. And I want to give you a bit of a history of the field, the context, some of the big questions we want to ask, and how ancient DNA is a little different, how we have to modify pipelines and workflows that are common in modern metagenomics to work with our ancient and degraded samples. The first thing I want to start, though, is what is a metagenome? Does anybody know? Anyone want to throw out a guess? What's a metagenome? I know you've heard this term before. You're in an ancient metagenomics course. Okay, people are shy. It's okay. Okay, we're warming up. There's more questions coming. So a metagenome is the collection of genomes and genes from members of a microbiota. All right, what's a microbiota? Okay, what's going on here? So this is the collection. This collection is obtained through shotgun sequencing of DNA extracted from a sample. That's metagenomics, followed by either mapping to a reference database or assembly, so de novo assembly, followed by annotation. There we go. And a microbiota is an assemblage of microorganisms that are present in a defined environment. And finally, a microbiome refers to an entire habitat, including the microorganisms, their genomes, and the surrounding environmental conditions. These terms were defined in 2015 by team Marchesi and Ravel in an article called The Vocabulary of Microbiome Research. And now you might be wondering, why did we have to have an entire scientific article dedicated to just defining these terms? And the reason is that prior to 2015, terms that were used about the microbiome, about metagenomics, about microbiota, it was like a Wild West. Terms were being used in different ways to mean different things. It was extremely confusing in the literature, and it led to a lot of problems in building research because people would use the same terms to refer to different things. And so they, out of frustration, one of the earliest articles that was actually published in the new journal that was founded called Microbiome to serve this growing field was an article defining the terminology of the field. And like I said, the terminology about microbes was and continues to be to some degree a mess. And so if you've been confused over the course of your education, you are not alone. It's getting a little bit better, but hopefully today we'll clarify some of that confusion. So one of the problems is the term metagenome was actually originally coined by Jo Handelsman in 1988, but she used it to mean something completely different from what we use it for today. The term metagenomics has been used to mean different things over time, and especially in the early days of next generation sequencing was occasionally used to refer to 16S rRNA amplification, something we now call metataxonomics. And microbiome is claimed to have been coined at least twice, each meaning something different. So either meaning a microbial biome, meaning a microbial community, which was defined in 1988, or a microbiota-ome, meaning the collective genomes of a microbiota in 2001. But the term microbiome actually appears in the scientific literature since at least 1894 in different places, in different contexts. So it's been used a lot, but often to mean different and inconsistent things. And so we needed this article about vocabulary to bring us all on the same page so that when we say metagenome, or we say microbiome, or we say microbiota, we all know and agree with what we mean. But this is really important because if you start to go into the literature before 2015, you may see these terms being used to mean different things. So just watch out for that. All right, so what is ancient metagenomics? Ancient metagenomics is the study of the collection of genes and genomes of the microbiota within a given environment or microbiome, plus all the other DNA that's been mixed in. So basically, it's all the DNA that you're going to obtain in a sample. The key point here is that in addition to the ante-mortem, meaning before death genetic material of any microbes that may have been present in life, ancient metagenomes almost always contain at least some post-mortem DNA from the tenatomicrobiome and the necrobiome. There's two separate terms here. Tanatomicrobiome means the microbes that come from your own body that proliferate after death and contribute to decomposition, whereas the necrobiome refers to external microbes from the burial environment or the soil that invade the body after death and contribute to decomposition. These are two separate terms. You'll see them both in the literature. In reality, you have both the human's own body microbes plus environmental microbes that contribute to decomposition. And more people are just referring to both with the term necrobiome. So ancient metagenomics is in many ways a lot like regular metagenomics, but in some ways it's a lot harder because other environmental microbiota of various ages are mixed in, and because the DNA itself is ancient and degraded. So we try to do all the things that you can do with modern metagenomics, but with some of these additional challenges. So I'm going to use a metaphor here to kind of help bring this into focus. So yesterday we talked about puzzles, and so I like to call this my worst puzzle ever, metaphor of ancient DNA. I love jigsaw puzzles, but I definitely don't put together 60,000 piece ones except in my work. And so if you think about a typical bacterial genome being about 3 million bases long, when it degrades over time, it fragments into pieces, and on average that single microbial genome will break into about 60,000 pieces. Now that's bad enough, but because we're working with metagenomes, we never actually have a pure single genome to work with. Instead of working with one puzzle, we have to work with lots and lots and lots of puzzles. So let's just use this puzzle metaphor. Imagine you have a piece of paleofeces from the past. That paleofeces will include a number of different things. It might include the human genome. It might have a Salmonella enterica genome that from an infection that person had. It's embedded within the paleofeces itself, which contains the microbiome, the gut microbiome. And then you're going to have some additional DNA, which is the necrobiome that's kind of invaded in and is kind of contributing to decomposition to some degree. And all of this is going to be your metagenome, all these things mixed together. So all these different puzzles. Now that would be bad enough, except one of the problems we face is not only do we have so many puzzles, but someone has thrown away all the boxes. So we don't actually know how many different genomes are there, of what kind, how many of each one. And in some cases, we've never even seen the boxes before, so we don't even know what it's supposed to look like. And all the pieces have been mixed together. So that is the challenge we face with ancient metagenomics. I'm going to use another metaphor here. So that was our puzzle metaphor, but another good metaphor is that of a coral reef. So when we talk about ecology or microbial ecology, people often use a metaphor of a coral reef, which is actually quite apt. So if you think about a coral reef, it's this complex ecology of many different species that all work together to form something bigger than itself. So a coral reef is really this kind of triumph of ecology, of multi-species ecology with fish and coral and all sorts of different organisms that all live together in a kind of harmony. And so you can imagine our ancient samples that we might be interested in, maybe our ancient dental calculus or our ancient paleofeces, as this beautiful coral reef full of all these microbial taxa all working together. But we have a problem, and that is time. This is a picture of a parrotfish. Does anyone know what parrotfish eat? They eat coral. So you can think of our parrotfish here as a metaphor for time. So you can think about this, our kind of coral reef being everything that's in the past. Our parrotfish is time, and it comes along and eats the coral, and then it poops out the ancient DNA, right? So that's the white sandy beaches that people love. That's actually parrotfish poop. That's the pieces of the ground up skeletal bodies of the coral. And that's kind of what we have to work with at the end of our project here. So I call this my parrotfish metaphor of ancient metagenomics. So here is our ancient metagenomic DNA, this pile of white sand. And we're going to use a number of techniques, both in the wet lab and computationally, to try to reimagine that original beautiful coral ecology. But all we have to work with is this pile here. So there's a lot of challenges that we're going to face. Now, at this point, you might be thinking, oh my God, I'm out. I'm so done. I'm not even going to start. But don't worry, there's a lot of tools that can help you. We can definitely do this together. All right, so the first question you're going to want to ask from your ancient metagenomic DNA is, who's there? what species are there? That's kind of always the first starting point. What organisms are present in this pile of ancient DNA that I've now generated? The next question you want to ask is, well, how preserved is my sample? Does it actually preserve the original microbiome or the original pathogen, or is it completely turned over and composted with decomposition bacteria and necrobiome? Then once you've determined the degree to which it's preserved, you want to clean up that data set. You want to remove the obvious contaminants and like the modern DNA to try to get something as close as you can to that ancient microbiome, that microbial community, and then reconstruct your beautiful microbial community from the past that you're interested in. And so we're going to start with these first questions today, and then later this week on Wednesday, you're going to learn how you can look for something very particular, for example, pathogens, so later today. Tomorrow, you're going to learn a bit more about how we can move beyond reference databases to do de novo assembly and build MAGs to explore the genomes that are present in your data set, both known and unknown. And then on Friday, you're going to focus more on authentication. Can I trust my data? How do I know that it's real? What are ways that I can show that I've really recovered a true ancient genome from the past? All right, so let's go back to those first couple of steps, which we're going to cover today. The first is, who is there? So at a most basic level, the first question we all ask in metagenomics is, who is in my community? But before we can do that, we really have to define what we mean by a microbial species. And where does the species concept come from? So the kind of modern species concept that we use in biology is largely based off of foundational work done by Ernst Mayr on the biological species concept, which he published in 1942. Now, he built this concept of the species off of his work on birds. So the kind of biological species concept that we use in biology is based off of a taxon of birds that's quite different than microbes. Fun fact, Ernst Mayr was the advisor of Jared Diamond, who later went on to write Guns, Germs, and Steel. In fact, you'll notice here that they co-published this book, The Birds of Northern Melanesia, together. All right. So we have to adapt this definition of species that Ernst Mayr defined for birds, which is, a species based on a sexually reproducing organisms that have a completely different life cycle than our small asexually reproducing bacteria. So we have to define what is a microbial species. And here I've shown a small phylogenetic tree for two groups of bacteria that are very common in the oral microbiome, so Tannerella and Porphyromonas, which you see here. And in order to define microbial species, we have to talk about two really foundational concepts, and that's taxonomy and phylogeny. So taxonomy is the classification or categorization of organisms into groups, which we call taxa. And phylogeny is an evolutionary history of a set of taxa. Now, ideally, when we want to name a species, we want these two things to agree. We want the taxonomy, the name we give to that species, to reflect its evolutionary history. But that can be sometimes hard to determine. In the case of Tannerella and Porphyromonas, what we can see is here this agrees. So the phylogeny and the taxonomy are in agreement. So all of the Tannerella are clustering together in one clade, and all of the Porphyromonas are also clustering together in a kind of a clade that then bifurcates, but it's coherent. It's phylogenetically coherent. So these are the taxonomy and the phylogeny in this case agree. But that's not true for all the microbial species that you're going to encounter, especially for species that were discovered and cultured and named in the 19th or early 20th century before they had robust methods for categorizing them. We often get kind of mixed taxonomy and phylogeny that are incongruent. And a really great example of that is this group here of Klebsiella, Salmonella, Escherichia, and Shigella. So these are quite important. They've been studied for a very long time because many members in this group cause gastrointestinal disease. But you can see here that actually this is a case where the taxonomy and the phylogeny do not align. In fact, Shigella, Escherichia, and Salmonella entirely fall within the diversity of Klebsiella. So if we were to use a proper taxonomy based on the phylogeny, those would all be subspecies, or they would be nested within Klebsiella. within this group, Salmonella does form a coherent clade, but look at Shigella, it doesn't. It's intermixed with Escherichia, and then some of them are even down in this kind of lower Klebsiella branch. So actually, it's all mixed up. So you might say, oh, this is a Shigella, it must be closely related to other Shigella, but in reality, it might be closer related to different Klebsiella or Escherichia than it is to other Shigella, and that just has to do with the fact that these different species were named long before their true phylogenetic relationships were understood. And there are teams of microbiologists that are combing through the literature and revising these taxonomies and updating the names of these microbes. So one thing you may have noticed, which makes our field quite difficult sometimes, is that a lot of our microbial species are being renamed. So just a couple of years ago, the entire Lactobacillus group was renamed and split into dozens of genera, and that was to reflect the fact that the phylogenies of this group did not reflect the taxonomy that was being used. All right. So when we talk about microbes, though, it's really difficult to apply kind of standard biological species concepts because of their very different kind of life cycles. And so in many ways, the species definition we have for microbes is fuzzy. It's a fuzzy definition. It's hard to define very precisely. We do put microbes within a Linnaean taxonomy, which you see here, and these are different levels of taxonomic classification. So at the most specific level, we have the species. So for example, this is Capnocytophaga gingivalis, which is a common oral bacterium. It's in the Capnocytophaga genus, it's in the Flavobacteriaceae family, it's in the Flavobacteriales order, it's in the class Bacteroidea, and in the phylum Bacteroidota, and in the domain Bacteria. And so this classification system, which you're probably familiar with from school, from high school biology, is a way to classify all the different organisms of life into their phylogenetic relationships. Now, to understand, though, how this system developed, we need to learn about the two Carls. Does anyone know who the two famous Carls in taxonomy are? It's all crickets today. Okay. The first is Carl Linnaeus. So I'm sure you've all heard about him. He invented the system of binomial nomenclature, the genus species naming that we use to name all living organisms on Earth. He started this in 1753. And fun fact, earlier this year, I got to go to the Swedish National Museum and actually see his original type specimens that he had handwritten those binomial names onto. It was very exciting. If you have a chance, go to Stockholm and see this museum. It's incredible. And so Carl Linnaeus is really important because he establishes the framework that was built upon to kind of systematize the relationships between different organisms. The other really important Carl is Carl Woese, who in 1990 coined the term domain to be a higher level taxonomic group for example, here, bacteria versus eukarya, defining these groups that are above the phylum, which was part of the kind of classical Linnaean taxonomy. Now, the problem, though, is because it's very difficult to define a microbial species, and because oftentimes we have to base it off of kind of less direct methods, maybe only a set of marker genes or various types of ways to infer different characteristics of these microbes, we often talk about operational taxonomic units rather than a species. So the species is kind of the ideal. It's our idea that there is a real relationship between these organisms and a relationship between their taxonomy and phylogeny, but that phylogeny may not always be known to us. And so instead, we use things that we can measure, which are imperfect metrics for that phylogeny, to try to divide them into groups. And so depending on which type of imperfect measure that we're using, the different groups that it creates, we can refer to as operational taxonomic units, sort of like roughly a species, roughly this group, not perfect, but as good as we can get at any given time with some of the methods that are available to us. And so that's what operational taxonomic unit is really referring to. So let's dig into a bit more of who's there. How do you go from your raw DNA sequences to your taxon tables? So typically what we do is we start with something called a taxonomic profiler, and there's many different kinds. You have many different available options that have different strengths and weaknesses. There are two main flavors of taxonomic profilers. One group is the alignment-based profilers. This is going to be things like CHIME for 16S RNA marker gene categorization, things like Metaflon, which uses a genome-wide marker set, MALT, which uses read alignment and bidding. All of these methods can be used to classify the different reads or sequences, either all of them or part of them from your sample. And then there's other options that are alignment free, and the most prevalent one used there is something called Kraken, or its kind of subsequent versions, which uses k-mer matching. So let's go take a closer look at some of these. Let's talk about some of the classic methods. So the classic method here is the 16S rRNA amplicon metataxonomics, or using that 16S rRNA gene, which is a universal gene within bacteria and archaea, to try to classify different microbes into these operational taxonomic units that are not exactly species, but provide us with some way of kind of speaking about them and their relationships. So it's based on amplifying up the gene that encodes the 16S ribosomal RNA gene, and 16S is always spelled with a capital S because S stands for Svedberg units, which is a unit that refers to the density or the kind of molecular size of this ribosomal protein, ribosomal RNA kind of protein RNA complex. This gene is ubiquitous amongst prokaryotes, so it's really handy for trying to classify all different archaea and bacteria using one system. The gene itself is about 1,600 bases long, and it has this really interesting pattern where there are portions of the gene that are highly conserved and nearly identical across the tree of life, and other parts that are extremely variable. And the reason for this pattern is that it encodes RNA that folds up into a structure that participates in ribosomes. And some parts of that ribosome are so critical for function that they effectively cannot change, otherwise they become defective, whereas others are more free to mutate. And so you can see here, which plotted the different variable regions along the gene and where they're located. And the higher the peak, the more variable they are amongst taxa. So some of them, like the V1 region and the V3 region, they contain a lot of diversity, which is really great for discriminating against between many different taxa, where something like V9 actually has relatively low variability. And so you're going to get less resolution in discriminating your taxa. And so what people have done in the past is they have used PCR to amplify parts of the 16S rRNA gene. And they use this gene sequence, which they refer to as a barcode, a taxonomic barcode, to infer the operational taxonomic unit, or the approximate species or genus that it's from. Sometimes people call this approach of metataxonomics metabarcoding. They mean the same thing. There's a lot of different tools that can be used to help with this. Things like some of the popular profilers include CHIME, but also Mother, RDP classifier. There's many available. There's also huge databases available for this. For example, the Silva database. There's a lot of comparative information. It's efficient and inexpensive, and it's really widely used in microbial ecology for modern DNA. And where this becomes important is that 16S rRNA sequences historically were what led Carl Woese to the 1990 discovery that archaea represented a new domain of life. Now, these microbes were known. These are kind of strange extremophiles that had been identified at the time in things like hot springs. But because they were microbes and they looked similar in some ways to bacteria, they didn't know how different they were until Carl Woese sequenced their 16S rRNA gene and found that they were really distinct from both bacteria and eukarya, thus discovering an entire new clade of life at such a fundamental level that it led to the invention of the term domain to distinguish between these groups. So we now have 3 known domains in life: the bacteria, the archaea, and the eukarya. There's a couple of famous archaea in this group that many of you might be familiar with. For example, Methanobrevibacter oralis and Methanobrevibacter smithii. These are archaea that actually live in the human oral cavity and in the gut. At first, archaea were thought to be extremophiles, only existing in really extreme environments like deep sea vents or thermal hot springs, but we actually now know they exist all over in many different environments, and there's quite a number of them that actually live in the human body. But one of the problems for using the 16S RNA marker gene approach for ancient DNA is the fact that it stems from the kind of biology of the ribosome itself. So of the different variable regions that I mentioned, the V3 region is the one that in many ways is most suitable for ancient DNA because it's the shortest variable region. Remember, ancient DNA is very short. So we can't amplify really long stretches of DNA. They simply aren't preserved. So we have to have a target that's short. And it has pretty good taxonomic discrimination. But big problem, it's actually length polymorphic. And you can see this over here on the right. So the way that the RNA folds up to function in the ribosome, it has these stem loop structures, and some of those stem loops are kind of freely able to evolve. So for example, in Methanobrevibacter oralis, it has a relatively short stem loop structure in the V3 region, whereas in other taxa like Streptococcus pyogenes, it's evolved to have really long sequences. And this causes problems because it means that our DNA template that we're amplifying off of is different lengths. And the V3 region itself is really too long for ancient DNA. It's on average about 180 bases long, and if you compare the sort of target length with the actual measured fragment sizes of ancient DNA, you'll quickly see that we're amplifying in the tail of the distribution, just the tiny portion of ancient DNA that happens to be long enough, which represents only a tiny fraction of the ancient DNA that's actually there. So this causes a lot of problems, and we've done a lot of studies on this that have shown that if you try to use the 16S rRNA marker gene amplification approach for ancient DNA, you're going to get a lot of biases, and really you cannot apply it to ancient DNA and generate usable results. Now, it is possible to comb through your metagenomic data and pull out those 16S sequences, but that's very inefficient because these 16S sequences represent less than 0.05% of the total DNA in your sample. And when classifying very short sequences from the 16S rRNA gene, it's quite error prone. You're going to have low taxonomic resolution, and you're going to have a lot of false assignments. And so now we recommend alternative approaches entirely using metagenomics. So the field of ancient DNA was actually transitioned very early, already in 2015, almost full-time into metagenomics. And so we built up a lot of expertise in this field. So sorry to say, at least for the purposes of ancient DNA, rest in peace, 16S gene. Let's next talk about the real workhorses of ancient DNA metagenomics, and that's going to be Metaflon, MALT, and Kraken. So Metaflon uses a marker gene set. It's a taxonomic profiler that uses short-read DNA sequence data and a database of marker genes that are highly specific to certain clades. So they've been curated to be maximally taxonomic discriminating. The marker database contains more than 1 million markers from bacteria, archaea, and microeukaryotes. And there's a number of different versions. So the first one, Metaflon, came out in 2012, then Metaflon 2 in 2015. Those are both now retired. Metaflon 3 came out in 2021, and now the current version that's being used is Metaflon 4, which came out in 2023. Metaflon 4 has the largest database. It includes 236,000 microbial genomes plus 771,000 MAGs that have been added. So it's quite extensive. And you can get it from the biobakery. Some pros is it uses metagenomic data, so it's using a lot, the full genome-wide data that you have, and it works really well with ancient DNA. It's computationally very efficient. The marker database that is used is good for pathogens and for the human microbiome in particular, but some cons are that because it uses a defined marker database, it has low customizability. And the marker database is missing taxa that may be relevant for other oral microbiome or other animal microbiomes or environmental DNA. So it's not going to perform as well on those samples. Also, it only profiles microbes. So if you want to also look at the plants or the animals that may be in, say, paleofeces, you're not going to be able to do that using Metaflon. But overall, it's a good option for human-associated ancient microbes and microbiomes. It was developed by Curtis Huttenhauer and Nicola Segata, and they've innovated many microbiome software tools, some of which you may have used before. Some of the other great tools that have been developed by this same team include PhyloFlan, which is for phylogenetic profiling of genomes and MAGs, PanFlan, which is for pangenome strain analysis, Human for functional profiling, And the team is also vastly expanding available microbial reference genomes through large-scale metagenomic assembly projects. You're going to learn more about this tomorrow. But for example, they released 150,000 reconstructed mags from the human microbiome in 2020, and then just a year later, another 200,000 mags. So they're building up this huge amount of reference data on the biodiversity that is entirely culture-free from these assembled genomes. The other tool that's widely used is MALT. This uses read alignment and binning. It was developed by Daniel Hewson and Alexander Herbig, who works here at our institute. It's a short read DNA sequence aligner for metagenomic data, and it's integrated into the Megan or the Metagenome Analyzer software suite, which allows the sort of graphical user interface of inspecting your data. The acronym MALT stands for Megan Alignment Tool, and it works really similar to BLAST, if you're familiar with that, but it's much faster. And it was developed as a DNA alternative to the protein sequence aligner Diamond for use in Megan. And the reason for that is when you're working with ancient DNA, you don't want to use software that translates your sequences into protein sequences for taxonomic classification. Your DNA is already short enough, and that process actually reduces the information in your data even further and will reduce your actual ability to have more precise taxonomic classifications. So we really have to work with the DNA sequences, not the translated protein sequences when profiling our ancient samples. So don't use Diamond or Kaiju or any of these other profilers that do this translation step first. That was really developed to speed up the computational process for really well-preserved modern DNA, but it will actually decrease the quality of your DNA if you apply it to ancient DNA. MALT has the advantage of using all of the DNA in a data set, so it doesn't rely on a marker gene set, but it just uses everything to perform taxonomic classification by aligning to a reference database, and you can choose it. So you can choose NCBI-NR or RefSeq, and this is great because it means you can go beyond microbes and also include those plants you're interested in or those animals or whatever. But this also makes it slow and memory intensive, but it does maximize all the data available. It is highly customizable and, like I said, can be used for all taxa, not just microbes. And then it uses a lowest common ancestor or LCA algorithm to assign each sequence to a node in that taxonomy, so you can tell how specific you can really be about the assignment. You may have Yersinia pestis in your data set, but maybe the read's very short, and so it can't be precisely aligned to that species, but it may place it on the Yersinia, the genus node, for example. All right. Some pros about MALT is it maximizes the use of your data. It has good database customizability, and it can profile all taxa in a sample, not just microbes. And you have the Megan tool interface for quick data inspection. And it's integrated into the NF Core Eager pipeline, and it's compatible with HOPS, which is a separate software tool that was designed to be compatible with it to do a high throughput pathogen screening. And because it produces alignments, you can easily create DNA damage profiles to authenticate your results. But there's also some serious cons. It is very computationally intensive with large databases. And as your databases get bigger and bigger, you really need more and more resources to the point where it can become impractical for your local setup, even on a high performance computing cluster. And it can be quite buggy. Next, we're going to talk about Kraken. Kraken uses a very different approach. It uses a k-mer matching approach. So it does not use alignment. And because it's using k-mer matching, this makes Kraken much faster and much less computationally intensive than alignment-based profilers. Its database is customizable. It can be used for all taxa, not just microbes. And this was developed by Derek Wood and Steven Salzberg in 2014. They've added on a number of new features and some additional tools in the Kraken family. So for example, you can now correct to account for genome size differences when calculating species abundance with the tool Bracken. You can also reduce false positives with their tool Kraken Unique. And in 2019, they released Kraken 2, which is even faster and more efficient than the original Kraken. Some pros about Kraken is that it is fast. It can be used for any set of taxa, not just microbes, and it's great for quickly seeing what's in your data. In fact, we basically use it as a first step to inspect our data when we're analyzing a new sample. Its accuracy is good enough for most ancient microbiome studies, but if there's a very specific species you're interested in, say a pathogen, then you need to do more work to validate it. Some cons, it can be prone to false positives, so watch out. You want to always go that extra step of validating after a crack and run. It does not provide alignment data, so you would have to separately perform damage analysis. All right, so let's compare some of these. We've done this a number of times. This is a paper we had a few years ago, led by Warinner, where we compared the performance of different classifiers on real-world ancient DNA datasets and also on simulated datasets to look at systematic biases that might be there. The take-home message is no taxonomic profiler is perfect. All of them have problems with false positives, but they tend to be primarily found in the low abundance taxa. So by removing singletons or really low abundance taxa from your data set, you can dramatically reduce the number of false positives that you have. Taxonomic profilers generally return broadly similar results, but they do have some predictable biases. And database selection will strongly impact the precision and the accuracy of your taxonomic assignment. So take-home message here is, which profiler do I use? It really depends on which question you're trying to ask. So what you want to do is select the best profiler for your particular study. The other thing I want to emphasize here is it's not just about the profiling tool. The database you use matters so much. Many databases are incomplete, and you won't be able to find what you can't see. So you have to always check to make sure your database contains the taxa of interest. So if you use a database that doesn't include the taxon you're looking for, you're never going to find it. So let's give it an example here. So the first Metaphlon database lacked the genome for Tannerella forsythia, which is a really important oral microbe in the mouth. And so this common oral microbe would just disappear from your analysis if you analyzed it with Metaphlon. The new Metaphlon 2 and 3 databases and Metaphlon 4 fix this. It's now included, so it is detectable. But in the beginning, we couldn't figure out why every time we profiled with Metaphlon, all of our Tannerella forsythia would disappear from our data set. So if your taxon is missing a reference genome in the database, your DNA might not just disappear, but it could align to the next best or the next closest thing, and this was what causes those false positives. This can be especially problematic because many pathogens have closely related environmental relatives, and they can often, those environmental relatives, if they're not in the database, can mismap their DNA to your pathogen, falsely looking like it's full of pathogenic reads when it's actually full of a harmless relative. Another example here is taxonomic profiling of dental calculus prior to 2012 always returned results that the pathogen Propionibacterium acnes, which causes acne, was prevalent and abundant. We saw in almost every oral data set so much Propionibacterium acnes. However, after the genome of the related oral species, Pseudopropionibacterium propionicum, was published in 2012, P. acnes just disappeared from all the data sets. And that's because actually P. acnes was never present in the oral cavity. It was always Pseudopropionibacterium propionicum, but because that genome wasn't included, those reads would map to the next closest thing, which was this skin pathogen. And then, just to make everything more confusing for all of you, P. acnes was later renamed Cutibacterium acnes in 2016, so you no longer see it in more recent literature. I think someone who was not a teenager thought they were being very cute when they gave it its new name. All right. The other thing to know about databases is they contain a lot of junk data. Databases are amazing. The Sequence Read Archive at NCBI and the European Nucleotide Archive, these are major human achievements. They are incredible, but they are crowdsourced data for the most part. Labs all over the world are contributing data, and there isn't a lot of checks or even the ability necessarily to check on the quality of some of that data. So people make mistakes. And so you should know these databases do have errors. So you always have to double check if you think you have an amazing finding. So genomes in NCBI contain errors, and even the reference sequence genomes contain errors, and sometimes big errors. So just a couple of examples here. Common carp, Cyprinus carpio genome. The people who uploaded that database, they forgot to remove the sequencing adapters. So the sequencing adapters are in the genome. So if you search your sample against that genome and you have any adapters at all within that are residual within your data set, it will show up as carp. And so a lot of ancient DNA data sets look like they're absolutely full of carp when really they're just residual sequencing adapters. Tibetan antelope, Panthelops hodgeni, turns up in every metagenomic data set. We're not entirely sure why. We think it's because there's microbial sequences that have accidentally been incorporated into its genome. And as a result, any soil bacteria that you might have in your sample will get identified as Tibetan antelope. And the RefSeq genome for the common soil bacterium, Achromobacter denitrificans, contains the entire chicken ovalbumin gene. That was a mistake. So there's always some errors in here, and you always want to just double check any extraordinary findings that you might make. Remember, your brain and your critical thinking skills are your best defense against bad databases, bad data, and wrong conclusions. So when in doubt, check and double check your work. All right, so we talked about our starting questions. We have our ancient metagenomic DNA. We've asked who's there. Whew, we've made it. Are we done? No, we're not done. Have a glass of water. We're going to go a little bit further. We're going to ask now, how preserved is my sample? So we're going to talk about metagenome composition and quality, and the causes of degradation and sources of contamination. The major sources of contamination, or kind of the DNA that is in your sample that's not necessarily your target of interest, it's not from the ancient environment or microbial community you want to analyze, come from the burial environment. So that's that microbiome, all those soil microbes that exist in those sediments who are very happily helping to decompose that ancient sample. We also get post-mortem microbial overgrowth. So there could be some of the, let's say you're studying paleofeces, and some of those bacteria don't immediately die. Maybe some of them actually grow and increase in frequency in the interval shortly after death, feeding on all the other microbes that are dying. And so you can have these shifts in the microbial profile from this post-mortem interval. And then you can also get microbes introduced through post-excavation handling and storage. Through the excavation process, by going to a museum. Museums are absolutely full of their own microbiome. And so sometimes those microbes can also invade and contribute to the DNA that's present in your metagenome. So what you want to do, though, is you want to try to remove as much of this as possible before you do your analyses. Otherwise, your analysis is mostly going to reflect these post-mortem exogenous bacteria and handling history, and not the original microbial community that you're interested in analyzing. And so there's a number of different techniques and tools that can help you characterize your data set's preservation state and potential contamination sources. We're going to talk about two processes that we use to clean up these data sets. The first is source tracking. And for that, we're going to use two different tools, Source Tracker and Source Predict. And there's some cleanup tools like CooperDeck and Decontam. All right. So microbial source tracking can be performed using a Bayesian or machine learning methods to estimate to what degree your data derives from a particular microbial source. So there's two main methods. So Source Tracker, which is developed by Dan Knights in 2011, or Source Predict, developed by Maxime Bory here at the MPI in 2020. So the user provides reference metagenomes. So let's say Human Microbiome Project dental plaque, feces, or soil metagenomes as sources. And then the tool tries to estimate the proportion of your data set that derives from one or more of these sources. So here's an example using Source Tracker 2 from one of James's papers, where we were looking at dental calculus from a variety of ancient humans and also historic collections of primates. And colored in red are sources that relate to the human oral cavity, so subgingival plaque or supragingival plaque. And then things like in the greens and blues are things like skin, environmental control, and sediment. And this is dental calculus that we're analyzing. So what you can see is that some of our samples are very well preserved. So the overwhelming majority of DNA in those samples is estimated to come from an oral source. Whereas we also have some samples that are very poorly preserved, are really estimated to have no oral DNA at all. And so those are some really poor samples for us. And so we can inspect the quality of our data to make some decisions about which samples we may want to include in our studies and which ones we think are too damaged and degraded to carry forward. But we often see this kind of gradient of preservation between very well preserved and very poorly preserved samples. And so where you draw that cutoff of what to include and what not to include will really depend on your own study's questions. Now, not all samples show that degree of variation. Here's another plot that comes, we're using Source Tracker again too. This is on more recent samples. That previous example included Neanderthals and quite old samples. This is more recent samples from the oral microbiome from 19th century Europe and medieval Europe. Here, the color palette is different. So here, blue and green actually are good. That means oral, and it's the kind of yellows and reds and grays that mean not oral. And here you can see actually almost every sample that we analyzed preserved, more than 75% of the DNA is estimated to come from an oral source. This is really high quality data. So all of these have excellent preservation. We also might want to go beyond just preservation and say, actually, what is my sample? And this became a big issue for us when we were looking at paleofeces, and we started to slowly realize that some of the human paleofeces we were working on probably wasn't human at all, what was actually dog poop. It turns out the archaeological record is full of dog poop. And so we needed tools and ways of discriminating between human feces and animal feces, and developed a tool called CopperID to do so. So some pro tips here: you want to choose your sources wisely when developing your source tracker analysis so that you can pick up the different sources and maximize the utility of the method. You're going to need at least 10 data sets per source for it to be statistically valid. Plaque and calculus, you should know, do have similar but slightly distinct profiles. Calculus has a more mature biofilm, so if you can include like modern calculus as a source, you'll get improved results. Also, when looking at soil or trying to model the microbiome from soil, it's often better to use sequences from archaeological bone than soil itself, because within human skeletal remains, only a subset of the soil microbes tend to thrive. And so rather than using soil itself, we often use, say, archaeological bone from the foot, which as our kind of proxy for environmental contamination. One important caveat to keep in mind, though, is that in this analysis, there's a category called unknown, and unknown here refers to two very different things. It's both the proportion of your data set that you can't assign to any source, but also the proportion of your data set that can be assigned to more than one source. So be careful when interpreting that unknown category. All right, so we've gotten through who's there and how preserved is my sample. Now let's talk about how we're going to clean it up. This is a two-step process. The first step we usually do is we just identify the samples that we think are too degraded to carry forward. They're so altered by time and decomposition processes that we really feel like there's nothing that we can do with them. So we can first identify and remove those very degraded samples using a tool called CupriDeck. This is something that James developed, and we can identify the ones that are just very poorly preserved. But there's always going to be those gradient ones where they do have preservation, but there's still some contaminants in there we want to remove. We can identify and remove low-level laboratory and soil contamination using the tool Decontam. So CupriDeck is used to remove those samples that are kind of beyond hope. And CupriDeck allows you to identify them so that you can remove them. So what this does is, this is a model here. So shown in blue are examples for subgingival plaque and supergingival plaque. We're here going to be looking at calculus. And then we have a whole bunch of controls. And what it's looking at is it's kind of plotting the cumulative decay curve of oral microbes. And you can see that blue means pass and red means fail. And so when we apply this tool to a variety of reference sources, we can see our oral samples pass and these control samples that are not oral all fail. We can then apply that to our dental calculus, and we can identify which ones retain enough oral bacteria that we think they're good enough to proceed with for analysis, but those that are in red are estimated to resemble controls much more than they do oral samples, and we might then exclude them for our analyses because they're just so heavily altered. For those ones, though, they're kind of in the middle, you can use Decontam. So these are for samples that are okay, but might have some stubborn contaminant taxa that you want to remove. The problem is if you leave them in your data set during your analysis, they could bias or skew your diversity patterns, could lead to spurious results or false conclusions. And so you want to try and pull them out if you can. Decontam can help you identify the obvious contaminants and remove them. And what you do is you provide this tool with your contaminant sources, for example, your data sets from your laboratory blanks or your archaeological bone, and it will go through and remove contaminating taxa from your data set. It actually works really well. All right, so we've gone through who's there, how preserved is my sample, how do I clean up my data set? Now you've got my microbial community. What can you do with your microbial community? You'll learn about this a little bit more today, but I just want to give you a little bit of a flavor for it. Within ecology, there are many ways to examine the microbial communities in your sample in order to better understand them. And the two most common ways that people start is by looking at their alpha diversity and their beta diversity. So the alpha diversity measures the variation within a single sample. Pro tip, however, just so you know, alpha diversity is very easily skewed in ancient samples by preservation and any trace contaminants you didn't manage to remove. So you have to be very careful if you're going to analyze the alpha diversity of a sample. Usually when people look at alpha diversity, they look at species richness using a tool like an approach like, for example, the Chow-1 index. And this basically answers the question of how many different species are in my microbial community. People also frequently look at species evenness using something like the Shannon index. And this really addresses the question of how balanced are the species in my community? Do a few taxa dominate a sample, or is it more evenly spread across many taxa? You'll see these sorts of alpha diversity plots in a lot of publications, including, for example, this one here from the Human Microbiome Project, which showed that oral samples actually have pretty high alpha diversity. Beta diversity measures the variation between samples. There's different ways you can do this. A common one is using Bray-Curtis dissimilarity, which addresses the question, to what degree are taxa shared between my samples at the same abundances? So if they're exactly the same, then you're going to get a score of 0. And if they're completely different, you're going to get a score of 1. So it's looking at how similar two different communities are. Another metric that is used is Jacquard distance. And this asks to what degree taxa are shared between my sample, but ignoring those abundances. Just looking at species presence absence. And then UNIFRAC asks the question of how phylogenetically similar are the taxa in my sample, and you can do this taking into account abundance, that's weighted, or not, so unweighted. This is nice because you may have many different, say, Streptococcus species, but they're all within Streptococcus. The other two methods won't notice that those are actually really similar organisms, but UNIFRAC will. Beta diversity plots look like this. You can visualize them. You can visualize the beta diversity of a given set of samples using principal coordinates analysis. And here's an example of one that's using Bray-Curtis distances from the Human Microbiome Project. And you can see that each one of these body sites has a distinct microbial community. And here's another example of something similar, but now on ancient data. And so what you see here, oh, thanks, James. In pink is our dental calculus and our plaque and our ancient calculus. Thank you so much. And our paleofeces is shown in various shades of yellow. And what you can see is that our ancient calculus really overlaps and our ancient paleofeces overlap within those kind of distribution of the modern samples. And in particular, the paleofeces overlaps with non-industrialized modern gut microbiome samples. You've never heard of PCOA, so you don't know what it is. So principal coordinates analysis, it's applied to your distance matrix, so either Bray-Curtis or Jacquard or UNIFRAC, in order to visualize your beta diversity in a plot. For those of you who are coming from other fields of biology, you may be more familiar with PCAs or principal components analysis. So you can transform your data to use PCA. So you can take an entirely different approach by transforming your data in your taxon table using centered log ratio transformation or CLR. You can then build a Euclidean distance matrix, which is also an Atchison matrix, and then perform PCA analysis. So you can visualize your data either using PCOA or if you transform it as a PCA. Which is better? This is a bit of a philosophical debate, and there are strong feelings on both sides. They are both valid for metagenomics, but with different caveats. So whether you use like a PCOA or a PCA approach, they represent your data in different ways. I suggest trying both. The main difference is they deal with zero count data in different ways, and they handle discrepancies in sampling effort differently. You can read a lot more about the pros and cons of using these two different approaches in this article by Glor et al. In 2017. And if you're intrigued and want to know more, Patch Loss, Who Created Mother, has a series of YouTube videos about ecological analyses and distances, and he explains in detail how to use the R package vegan for a lot of microbiome analysis. I highly encourage you to check those out. He does a great job of explaining it. All right, so what's next? We're going to be later in this week, you're going to be learning about how we're going to apply now what you've learned today in a practical way. And hopefully then be able to start classifying your ancient data sets and making sense of this past microbial ecology that you have discovered. If you want to read more, I put the references for everything we talked about in this lecture at the end of the lecture slides, and I'm happy to take questions.